Hi, my name is Julia Engelmann. I'm from the NIOS, the Dutch Institute for Sea Research. Thanks for having me here today, and thanks for staying around in this last session. This is awesome. So I would like to tell you about the spatial and temporal variation of marine microbial communities and interactions around the West Antarctic Peninsula. Before we delve into this, let's take a few seconds to travel to space and look at our planet from space. And we can appreciate that actually the majority of the surface of our planet is covered by ocean. So it's a bit surprising that this planet is called Earth and not ocean. But anyways, what we cannot appreciate from space is that most of the life in these oceans is actually microbial, and that is both in cell numbers and in biomass. Now, these microorganisms in the oceans, they are responsible for about 50% of the oxygen that we breathe. So thank you, microorganisms. And for about 50% of primary production, that is producing organic material that then feeds into higher trophic levels, feeding the marine food webs, but also feeding food webs on land eventually. Now we're going to zoom into the Southern Ocean, which is particularly important because there's a lot of primary production happening in the Southern Ocean. And this organic material is distributed across the whole planet because of the currents that are generated by the Southern Ocean currents that circulate around Antarctica, but then also are connected to the other ocean currents. At the same time, Antarctica is exceptionally hit by climate change. Temperatures are rising there more dramatically than in other places on Earth. And we are interested in how this might affect microbial communities and then subsequently marine food webs. So quite a while ago already, we set out for a coordinated sampling effort, which is quite challenging if you're working on Antarctica. That happened at 2 research stations on the West Antarctic Peninsula. And they are indicated there with these red dots. So one is the British Antarctic Survey, the British Antarctic Research Station. And the other one is the US station, Palma. So the British station, I will refer to Rotorua, which is the place of the station. And the US station is Palma Station. So we have about 100 paired bacterial and microbial eukaryotic profiles. And in this little video, you can see how colleagues are taking a water sample with a Niskin bottle at Rotorua Station. So it's just a small rubber boat. And yeah, taking a sample there. Because this is also a long-term monitoring site, we have nutrient data and environmental parameters also available. Now I want to go to the next slide. Here. So don't be scared. We're just going to look at these community profiles really from a bird's eye view. And what you see on top is on the left corner is the Rotorua bacterial profiles. And on the right side, the Palma bacterial profiles. When you just look at this from a bird's eye view, you might appreciate that there's some similarity in these profiles, but there's also quite a lot of variation, especially in the summer. And this is because even though these 2 stations are both located on the web, they're relatively close, 400 kilometers apart. There are differences in temperature and salinity that lead to a different seasonality of phytoplankton blooms that are then followed by bacterial blooms that recycle the organic matter. So there's some variation in the summer especially. And the same is true for the eukaryotic profiles that are in the lower panel on the left side, again, Rotorua, and on the right side, Palma Station. And you see that at Rotorua, yellow thingies coming up, and these are actually parasitic groups that come up these are dinoflagellates that come up in the summer at Rotorua. And there's another group of parasites. These are in purple, more in the winter and spring. You might not be able to see them, sorry. This is another parasitic group, Syndiales, and they're not so abundant, but we'll come back to them later. And at Palma, we see mostly mixotrophs and autotrophs coming up in the summer. So these are the typical phytoplankton blooms that we are familiar with. All right, but I promised in my title that we are going to talk about microbial interactions. So the first thing that might come to mind is to look at correlation of abundances to predict interactions, but we're also very well aware that correlation has a lot of correlation-based methods have a lot of false positives. So the question is, can we actually do better and generate networks that have fewer spurious correlations that we will use as predictions? And can we maybe even infer a direction of an interaction? And we actually can. So I don't have enough time to go through this in detail. So just see this as a teaser. And if you're interested, then look up this paper and the references there and where it's explained in more detail. So causal effect estimation, in very short, works as follows. You first reconstruct a graph with the PC algorithm. And that starts with a fully connected graph. So we are now on the left side, sorry. So it's with a fully connected graph, and we're testing away edges that are not significantly correlated. Okay, that's not so exciting yet. But then comes kind of the magic part, which is on the right side here, that is able to infer a direction for some of the edges. And this is for cases where the correlation of two variables, in this case A and B, is depending on the status of a third variable, in this example C. And this gives us information about the information flow. So in this example, A and B are correlated whenever the status of C is low. And this is indicated by these blue crosses in the little data graph. And they're not correlated otherwise. Okay. That is maybe a bit abstract. So here's an example. So imagine a college, C, that accepts students who are very sporty, A, or very intelligent, B. So if you know that a student has been accepted to this college, then, sorry, just interrupted my flow. If you know that a student has been accepted to the college, then you know that the student must be either very sporty or very intelligent, right? So that gives you information. The second step is estimating causal effects using that graph that we just generated. And the causal effect is just a linear regression of the explaining your node of interest, which is Y in this case, with the upstream nodes in the graph, in this case B3 and the parents. So the causal effect of B3 on Y is the linear regression coefficient of B3 with also the upstream graphs, the upstream nodes in the regression. All right, so here's a teaser for the results, and there's a lot more results in the paper that I'll show you the QR code in a second. So this is a summary of the strongest effects, and we have them for Rotorua and Palma for two different size fractions. And what we can see is that there's a lot more of these greenish bars on the left side, which are eukaryotic effects. And they are more abundant at Rotorua and relate to the parasites, especially the parasites that are more abundant in the winter and not the very abundant ones in the summer, interestingly. Bacteria had more effects at Palma. And on the right side, you see the effects of environmental parameters. And here we could see that temperature was more important at Palma. And here's an interesting fact. Palma has been warming more than Rotorua, and also a lot more above freezing, above thawing temperatures. So that might explain why there's a large impact of temperature. So if you want to read more about this, please do check out the paper that came out recently. And with that, I would like to close, thanking Swan, the first author of this study, and all the other colleagues that were involved, including my colleague Linda from NIAS. And thank you.
Thank you so much. Any questions for the speaker? Okay, maybe I will ask a question. So with this approach to the network analysis, so how many samples do you need, and what kind of samples before you start to see something that's solid?
More than you have.
Okay. All right.
No, the practical answer is 30 plus.
Oh, I have more than 30, but not. All right, thank you.
All right, thank you.
I think now let's thank the speaker again. And